All right, you are all welcome back once again to the second lesson in our structures and function course in biochemistry. All right, so today we are going to talk about marker enzymes. Okay, that's our next topic after homogenization and self-fractionation. So we are going to treat marker enzymes, and learning objectives for today are going to be what are marker enzymes. All right, so we are going to talk about the definition for marker enzymes, what marker enzymes are, their characteristics, their useful purposes, and each and every marker enzymes and their organelle that are specific to it. Okay, so we are going to talk about marker enzymes for the various subcellular fractions called organelles, and after that we talk about the importance or the useful purpose of marker enzymes in the cell. Okay, so each cell has organelles, and each of the organelles can be identified by marker enzymes. All right, so we are going to talk about marker enzymes into details, what they are, each and every organelle and their marker enzymes, and after that we talk about the useful purpose, what marker enzymes are used for in the cell. Okay, so if you haven't watched our first video in the structures and function, you have to follow the link in the description below so that you can be able to understand what marker enzymes are. All right, so the first video was about homogenization and self-fractionation. And before you understand marker enzymes well, you have to know what homogenization and self-fractionation is so that you can get the maximum understanding from this marker enzymes video. Okay. So, what are marker enzymes? All right. So, marker enzymes, what are they? So, usually the cell contains organelles which perform varieties of functions. All right, so when we take the cell, for instance, we have the nucleus, we have cytoplasm, we have the chloroplast, we have lysosome, we have Golgi bodies, we have cytosol, we have peroxisomes. Okay, and each and every organelles of the cell perform various functions and different functions. Okay, so in order to obtain the precise information about the structure and function of this organelle, it is necessary to isolate them from other organelles. So if we want to determine the structure and function of, let's say, the nucleus, we have to separate it from other organelles such as mitochondria, endoplasmic reticulum, ribosome, so that they will not interfere. All right, so when you want to, let's say, determine the function of the nucleus, and there is a presence of mitochondria in it, I'm not sure you are going to get exactly what you want, because there will be a fraction of mitochondria which will perform a function and it's going to interfere with the function of the nucleus that you want. So in order to avoid this, we separate it from each other so that when we want to determine the functions of the nucleus, we will able to study it exactly and efficiently as how we want it. Okay, so hence centrifugation technique is done to separate them individually after homogenization. So that's why I talked about the first video. So we have to follow the link to watch so that you get understanding of what homogenization is and what centrifugation is in relation to marker enzymes. All right, so when to study marker enzymes of the various organelles, you have to get a homogenate, and before you can get homogenate or the cell lysate, you have to perform homogenization and centrifugation so that we can separate these organelles into pellets. Okay, so the larger ones will form pellets at the bottom and the lighter ones will form supernatant so that we can recentrifuge it to isolate them completely. Okay, so these individual organelles are identified using marker enzymes. So after homogenization and centrifugation, we can use these marker enzymes, okay, to identify whether there is a presence of, let's say, a nucleus, a mitochondria, or any types of the organelles. All right, so what are marker enzymes really? So marker enzymes, they are enzymes that they are exclusively and predominantly present in a specific organelle. Okay, so when we take nucleus, the marker enzyme for nucleus is specific, it's exclusive, and it's predominant in that nucleus, and it cannot be found anywhere else in the cell. Okay, so that's the marker enzyme of the nucleus will only be found in the nucleus. Likewise, other organelles. So it's same as cytosol. When we take cytosol, lactate dehydrogenase is specific for cytosol. Okay, and when we take the chloroplast, ribulose biphosphate carboxylase is specific for chloroplast. Okay, so that's how marker enzymes are and what they can be used for. Okay, so let's talk about our next slide. Okay, so marker enzymes for the various subcellular fractions, that's the organelles. So we are going to take each and every organelle one by one, and we are going to talk about their marker enzymes. All right, sure. So let's take the nucleus first. So the nucleus is in the cell that perform various functions. It controls hereditary materials, and it controls all the activities of the cell. So in order to identify the function of nucleus, what you have to do is to determine whether nucleus is present in a particular fraction or a homogeneity. Okay, and what we can use to identify whether the nucleus is present is the marker enzyme for nucleus, and that is nicotinamide nucleotide adenine transferase. So this is specific to nucleus. So after we've gotten the homogeneity and we've centrifuged it to get our pellet, and we want to confirm whether there is a nucleus present, we have to use a test, a reliable test, so that we can confirm boldly that there is a nucleus present. And this is done so by using the marker enzymes. When we do the test and we got nicotinamide nucleotide adenine transferase, then we can confirm that there is a nucleus present. So let's talk about plasma membrane. So the marker enzymes for plasma membrane is hormone stimulated adenylate cyclase. Okay, hormone stimulated adenylate cyclase. So this is the marker enzyme for the plasma membrane, and it is specific to plasma membrane only, and it cannot be found anywhere else in the cell. Likewise, mitochondria. So mitochondria has a marker enzyme called succinate dehydrogenase. Okay, and this marker enzyme is also specific and exclusive to mitochondria only. So we cannot find succinate dehydrogenase in the nucleus. Likewise, we cannot find nicotinamide nucleotide adenine transferase in mitochondria. It's not going to happen. Okay, so they are specific. So let's say endoplasmic reticulum has glucose 6-phosphate, and it cannot have succinate dehydrogenase. So that's marker enzymes for you. They are specific. All right, so let's talk about chloroplast. So it has ribulose biphosphate carboxylase, that's Robisco for short. Okay, so that's the marker enzyme for chloroplast. And the next one is Golgi complex or Golgi bodies. The marker enzyme for Golgi bodies is thymine pyrophosphate. Okay, so thymine pyrophosphate is specific to Golgi bodies, and after running centrifugation, we can use a test. So when we get a high amount of thymine pyrophosphate, then we can conclude that Golgi bodies is present in that fraction of our homogeneity. Okay, so let's continue with lysosomes. So we have ribonuclease for lysosomes, and we have catalase for peroxisome, and finally we have lactate dehydrogenase for cytosol. Okay, so lactate dehydrogenase is the marker enzyme for cytosol. All right, so these are the various marker enzymes for the specific organelles in the cell. So like I said earlier, there is not going to be a situation where you can find nicotinamide nucleotide adenyl transferase in cytosol. It is only specific to the nucleus. All right. Likewise, you can't find lactate dehydrogenase in the nucleus. It's also specific to cytosol only. Okay, so the cytosolic fraction is only composed of lactate dehydrogenase as a marker enzyme. So these marker enzymes are very, very important because they are used to identify these enzymes, these organelles in a fraction of a homogeneous, all right, so that we can study the functions of this organelle efficiently. Okay, so let's go to the useful purposes of these marker enzymes. So this slide is going to talk about what the importance of marker enzymes are. All right, so like I said earlier, isolation of organelles require a reliable test for the presence of the organelle. So when we have any organelle, it requires a reliable test so that we can identify the presence of that organelle. Okay, so in order to determine whether a particular organelle is present, we have to do a test. And before you can do that test, you have to homogenize it. All right, and this is done by using a blender or a sonicator, or usually a high pressure, as it was said in the first video. After that, you do centrifugation, density gradient centrifugation, to separate it according to their sizes. Okay, so there's the larger ones will settle at the bottom and they will form pellets, and the lighter ones will be at the upper portion called the supernatant. Okay, so the supernatants are rerun so that we can separate all the other organelles completely. Okay, so there is a link in the description. You can use that link to watch the first video which talks about homogenization and cell fractionation. Okay, so since marker enzymes, they are not to be found just anywhere else, their detection can indicate the presence of a particular organelle. Okay, so like I said earlier, marker enzymes, they are specific and they can only be found in just a particular organelle. So when we take the nucleus, for instance, we have nicotinamide nucleotide adenyl transferase, and this marker enzymes is only present in the nucleus. So its detection can indicate that there is a particular organelle called what? Nucleus. All right, and whereas their absence means the lack of a particular organelle. So when we get nicotinamide nucleotide adenyl transferase, then we can assume or predict that there is what? A nucleus. And we can likewise predict that there is absence of what? Mitochondria, plasma membrane, lysosome, peroxisome, Golgi complex, and what? Chloroplast. Since they are all absent and they can't be found in those organelles, then we can conclude that the organelle present is the nucleus. Okay, so their absence to defines the lack of an organelle. Okay, so marker enzymes also provide information on the biochemical purity of fractioned organelles. So these fractioned organelles are the homogeneous that we talked of. So before you can get these fractioned organelles, you have to go through homogenization and cell fractionation. So the cell fractionation is what we got these fractioned organelles. So the pellets are the fractioned organelles. So the pellets contains the organelles that are larger in size. So they settle at the bottom, all right, so that we can get these organelles from them. So when we get the pellets, then we can determine the biochemical purity by the amount of marker enzyme present in that organelle. Okay, so let's say for instance we did homogenization, and after that we continue to do density gradient centrifugation. We got our pellet and we want to determine whether a particular organelle is present. Okay, so we did the test, a reliable test as said here, and we got let's say 95% of lactate dehydrogenase. Okay, so lactate dehydrogenase is the marker enzyme for cytosol, the cytosolic fraction. Okay, so that means we can we can assume and presume that the cytosol is present. By what amount? That's the percentage purity. Okay, so that means there is 95% purity of lysosome in this fractioned organelle. Okay, so that's the important useful of marker enzyme. They are very, very important, and they are for identification and specification purposes. Okay, so before you can study organelles, their function, you have marker enzyme plays an important role. Okay, because you can't avoid marker enzymes to study the structure of organelles. You have to test first whether there is a particular organelle present in a homogeneous or a tissue. Let's say you have a liver tissue or a heart tissue, okay, and we want to determine the presence of organelles in this cell or this tissue. Then we have to do homogenization, centrifugation, and use the marker enzymes to predict whether this particular organelle is present. Okay, so for biochemistry, they can ask question like deduce how or write the process of how you can deduce or separate all the organelles from a heart tissue. Okay, so write down the process of how you can separate all the organelles from a heart tissue, a muscle tissue, or a liver tissue. Okay, it's a question that usually comes in the past questions for structures and functions of biomolecules. So you can try this yourself and write it in the comment section so that we can talk about it, and if there is any problem we can solve it in the comment section and you are free to go. Okay, so take notes of this marker enzyme very, very seriously. It usually comes in the past question. Okay, so the question that usually comes is write down how or explain how you can separate all the organelles from a muscle tissue or let's say a liver tissue. Okay, so it's very, very crucial part of marker enzymes. All right, so if you don't understand any part of the slide, any section of the slide, you can comment below and we will explain everything to you. If you have any question too, you can ask, and you have any suggestions too, you are highly appreciated. You can comment so that we will see the way forward. All right, take very good care of yourself, and the next slide or our next video is going to be stereochemistry. So in biochemistry, biomolecules, they are very, very specific. Okay, they are stereo specific. So before biomolecules can function, they need to be specific. Okay, so everything about biochemistry is specificity. So they are very, very specific in their actions, because biochemical nature is very important to the body. All the process that goes inside the body deals with biochemicals, and this process needs to be specific so that there can't be unforeseen circumstances in their reactions, because any reaction that is misappropriate can lead to a death or any circumstance. All right, so our next video we are going to talk about stereochemistry, and we are going to talk about chirality, optical activity of biomolecules, and we talk about optical isomerism, chiral synthesis, and other things. So stay tuned, and in order for you to get all the videos, what you have to do is just to like, comment, and subscribe to our channel. Okay, so subscribe, like, and share this video so that you get notified when every video is updated. Okay, so we promise to bring to you all the videos in biochemistry, structures, and functions. All right, so don't forget to subscribe again, like, and share. Okay, thank you very, very much.